Dear students, in this lecture we will discuss about the heme proteins, and this is the part 2 of heme proteins. We have already discussed about the general introduction of heme proteins in the previous lecture. So in this lecture we will discuss especially on the cytochrome P450. So now we will discuss about the cytochrome P450, which is a heme protein. This P40 means P for pigment and 450 means its absorption maxima in its absorption spectra lies at 450 nanometer. Number two, you can see that I have written as monooxygenase. So the cytochrome P450 is a kind of enzyme that is monooxygenase. So first of all, we will discuss what are oxygenase. So oxygenases are the enzyme generally which can insert either one or both oxygen atoms that are derived from O2 into an organic substrate. In this category, there are two kinds of enzymes. One is monooxygenase and other is dioxygenase. So in case of monooxygenase, it can catalyze the insertion of one oxygen atom into the organic substrate while the other oxygen atom get reduced to H2O. Okay, and when we compare this monooxygenase with the dioxygenase, then we will find that in case of dioxygenase, it catalyzes the insertion of both oxygen atoms into the organic substrate. So the cytochrome P450, it generally acts as monooxygenase because it can insert one of the oxygen atom of O2 molecule into the organic substrate. So first of all, we will discuss about the location of this cytochrome P450. So where it found in the human body. So it found throughout the human body, and the maximum amount is present in the liver, so that it can helpful in the metabolic processes, and in the liver, in the mitochondria and endoplasmic reticulum, it is found in abundance. So this cytochrome P450, its maximum concentration is observed in the liver, and in the liver especially in the mitochondria and endoplasmic reticulum, where it can helpful in the metabolic processes. Now, if we see the structural aspects of this cytochrome P450, then we will find that in the prosthetic group of this heme protein is iron third center that is incorporated in the porphyrin ring of heme B. That is it is it can be understand that this the prosthetic group of cytochrome P450 is a heme B group, in which you have seen that the substituent on the pyrrole rings are methyl, vinyl, and propionate, and in between in the cavity that is formed in between the porphyrin ring, Fe is present in the form of Fe3 plus. Okay, so this is the difference from the myoglobin. One difference is that here the iron is in its plus 3 state, while in case of myoglobin, iron is present in plus 2 state. The porphyrin ring is the same, but there may be the difference of the axial position ligands. Now you can see the cysteine thiolate residue is present at one of the axial position, where in case of myoglobin, histidine protein is residue is present at one of the axial position. So one ligand is cysteine thiolate here. The second difference is the iron third ion present, and one site is for the oxygen binding. So one is the oxygen binding site because this iron has 6 coordination. So four coordination with the four nitrogen of the pyrrole rings, one with the cysteine thiolate residue, and one is for the oxygen binding. Okay, and in the resting state, the ground state, the low spin Fe third plus is present because generally the oxygen binding site, it has the water molecule in the resting state. And when the oxygen molecule comes to this cytochrome P450, then this water molecule will replaced by the oxygen. Okay, so this in the resting site state, it has low spin Fe third center, and on binding the substrate it becomes high spin Fe2 state, and which non-cysteine ligand is displaced. Which is the non-cysteine ligand? The non-cysteine ligand is water molecule. So water molecule will be replaced by the oxygen containing substrate. Okay, so oxygen containing substrate is there. Oxygen binding site means the substrate having the oxygen center, it can bind, or oxygen can bind directly to this H2O molecule site. Here the substrate is actually the oxygen. So this oxygen, when it binds to the Fe3 plus low spin center, then it will replace the H2O, that is non-cysteine ligand. Okay, now this Fe2 plus, this becomes the from low spin Fe3 plus, it becomes Fe2 plus high spin complex, and this Fe2 high spin complex can bind with O2 to form oxyform, and with carbon dioxide it form carbonyl form. So it can bind with O2 or it can bind with CO2. Okay, and then this Fe2 oxidize to Fe4 on binding with O2. So on binding with O2, this Fe2 will oxidize to O2. That we will see in the mechanism of the reaction of cytochrome P450. This is the geometry of cytochrome P450 heme protein. It is an enzyme. This is monooxygenase enzyme. Here you will find again, this is the iron center, that is in 3 plus oxidation state. 4 coordination, they are through they satisfied through these 4 nitrogen centers of the pyrrole ring, while the fifth one is attached to the cysteine residue of the protein, and the sixth position of the ligand is H2O, that can be displaced when O2 will add to this center. And this red colored structure, this square, is the protein molecule. So this is surrounded by protein, so that it is altogether called as heme protein, and it is cytochrome P450. Now in this structure you can see this is the diagram of cytochrome P450, where you will find that here is the heme portion. So this is the heme portion where the porphyrin ring and either third center is there, and these helix, they are the helix of the protein molecule. So this is in whole, it is called as the cytochrome P450, that is a protein. Now come to the biological role of cytochrome P450. So what is the biological role of cytochrome P450? We will discuss in this slide. So cytochrome P450 enzyme, which is a hemoprotein, it plays an important role in intracellular metabolism. So I have told you that it acts in the metabolic processes, so that it finds in its place in the mitochondria and endoplasmic reticulum of liver. Most of the concentration is present. So in the intracellular metabolism, it takes parts in the metabolism of steroids, fatty acids, and xenobiotics. So what are xenobiotics? Xenobiotics are generally the external The chemicals that are generated by the human beings, and in the high amount they causes pollution. So these are the xenobiotics. So the cytochrome P450 enzyme can metabolize these xenobiotics, fatty acids, and steroids. This enzyme is present in prokaryotics and eukaryotics, including plants, insects, fishes, mammals, as well as microorganisms. And this acts as the protective mechanism in these organisms. Different kind of P450 enzymes can be found in almost all tissues, including liver, kidney, lungs, even in brain. And it is also helpful in the metabolism of drugs. So in this way, this cytochrome P450 is helpful in the detoxification of harmful substances also. So it plays an essential role in the biosynthesis, metabolism, and the detoxification of the harmful substances. The reactions by cytochrome P450, they include generally the oxidative reactions. So you can see in this reaction the carbon hydroxylation takes place, and you can find that the one oxygen of this oxygen molecule, it get inserted in between C and H bonds, so that it will convert this RH to ROH. So this is carbon hydroxylation process, and it is monooxygenase enzyme because it can add up one of the oxygen atom from the oxygen molecule to this organic substrate. So, thus, this organic substrate can be converted into a substrate that can be easily that is actually water soluble and get excreted from the body through the urine. So generally the cytochrome P450, it acts as the important role in the detoxification of harmful substances, so that it can convert these harmful substances into the water soluble substances, so that they can easily excrete out of the body. So in this example, you can see the oxidation of this aromatic ring. Actually, aromatic ring, no, this is the oxidation, hydroxylation of aromatic ring, and this O is inserted in between the CH and H bond. So this reaction takes place in the presence of cytochrome P450. This is the example of epoxidation of alkenes. So epoxidation of alkenes can also be helpful in uh, the conversion of insoluble this uh, harmed material to the soluble materials. Again, in this case, the sulfur get oxidized. So this is the sulfide oxidation, while this is the case where oxidative dealkylation of heteroatoms takes place. So these are the different reactions. Besides these reactions, there are a number of reactions that are catalyzed by cytochrome P450. As I had already told you that this cytochrome P450 is helpful in the metabolism of it. It plays an important role in the metabolism of steroids, fatty acids, and xenobiotics. So these reactions are the example of different reactions that are taking place in the metabolic processes of cholesterol. So you can see in this reaction, likewise, the 17 alpha, this carbon can hydroxylase. So you can see here OH is attaches here. Likewise, you will find this carbonyl group is converted to the OH group, and here you can find the conversion of this O to OH group. Okay, so there are a number of conversions can takes place in presence of a cytochrome P450. Likewise, you can see here at this position, this CH bond can be converted into this this OH, the H and C can be converted into COH. You can see that most of the cases, the converted compounds are generally water soluble alcohols. Okay, so insoluble aliphatic and aromatic hydrocarbons, they are converted into soluble alcohols so that they can easily be excreted out of the human body. This is the mechanism of the action of cytochrome P450. So this mechanism is actually start from here. So this is a molecule, this is the resting enzyme, okay, where you will find the iron is present as 3 state, and it activates the addition of O2. So it is the resting state of this enzyme. In this state, it can also bind any organic substrate also. So it can easily bind oxygen molecule, or it can bind the any organic substrate. Now you can see that this resting state can activate, we have already told that it can activate the addition of the organic substrate or organic molecule. So this hydrocarbon substrate, it binds to the enzyme, okay, and it the in its active site, and it induces the release of water molecules. So the water molecule will be released from here, and this organic hydrocarbon, it attaches here. The binding of RH to this center, it changes the spin state of the iron center. So the spin state changes from low spin to high spin, and this will increase the reduction potential, and one electron can thus transfer from this NADH, okay, and the biological, that is the biological electron transporter. So this NADH can transport its electron, and due to this process, a 5 coordinated Fe2 plus will generate. You can see that this Fe3 center can be reduced to this Fe2 center, and here it is 5 coordinated. Here also the iron is 5 coordinated, but due to the transfer of electron from NADH, this iron center will reduce to iron second center, that is 5 coordinated. This 5 coordinated Fe2 center, it resembles with the deoxymyoglobin. So if it is resembling with the deoxymyoglobin, then it can easily add up one oxygen molecule. So oxygen molecule can easily attack here. Likewise, CO can also attack in this center, and O2 can also attack at the center. So O2 can attack as it attacks on the deoxymyoglobin. Now you can see that this Fe3 will become, now this Fe2 will converts into Fe3 plus, and this Fe3 now it is 6 coordinated complex, and after formation of this complex, the second reduction will takes place. Okay, so second reduction will takes place, and this reduction leads to the cleavage of OO bond. Okay, so this second reduction, and after protonation, so the electron will add up, and this H2 will add this H plus will add up. So the protonation and reduction, so this reduction will helpful, it leads to the cleavage of this oxygen oxygen bond, so that one water molecule can be formed. So 2H and one oxygen from this oxygen molecule, it they come. So this reduction and protonation, it leads to the cleavage of oxygen oxygen bond, so that the formation of H2O takes place. Okay, and during this reduction, this iron third will convert into iron 5 complex, and this is an iron 5 oxo complex, where you will find one oxygen is double bonded to this iron, and at the one coordination site RH is attached. Okay, so this is a highly unstable species because of high oxidation state of this iron center. So due to high oxidation state of this iron center, this complex become very unstable. So due to instability of this becomes very reactive. Okay, and it becomes sufficiently oxidizing. Okay, so that this can oxidize the RH. Okay, so it can easily oxidize RH within the active side of the enzyme and give the product ROH. So this RH and this O, they will react to form ROH, and then regeneration of enzyme will takes place. And this regeneration of enzyme, it involves the uptake of hydrogen molecule at the vacated coordination site. So this is the complete cycle of mechanism of cytochrome P450. So I can revise again. This is the resting molecule, and here you can find Fe3 plus state, and this can easily bind organic substrate or oxygen. So during the binding of RAH, this H2O will evolve, and this state can easily reduce by taking electron from NADH and convert into Fe2 plus, which is 5 coordinated, and this is similar to the deoxymyoglobin, so it can easily add up oxygen molecule. So due to addition of oxygen molecule, its reduction and protonation takes place, and which leads to the cleavage of this OO bond, and due to cleavage of this OO bond, one water molecule will form, and this will convert into Fe5 species, that is an oxo complex of Fe5, which is very reactive, very unstable due to its high oxidation state. So at this stage, this ROH will react with this O, and thus it can form ROH, and this will again regenerate the enzyme with the evolution with the addition of H2O. This H2O will bind on the sixth coordination site of this cytochrome P450. So in this way, it can act upon the hydrocarbons. Now, concluding the lecture, in this lecture, we have discussed about the heme protein, that is an enzyme, and that is cytochrome P450, and that cytochrome P450 is a monogenous enzyme. So as this is a monogenous enzyme, it add up one oxygen atom to the organic substrate from the oxygen molecule, and we have studied about its structure, its location, where it is found in the body. Its main function is the metabolism of steroids, fatty acids, and xenobiotics, so that it can protect the human body from the harmful substances, because it converts these insoluble substances into soluble substances that can excrete from the body easily. Then we have discussed its structure, very easy structure, and the structure resembles with the oxymyoglobin, myoglobin. The difference is that in case of cytochrome P450, the iron is in its +3 state, and fifth coordination takes place through cysteine. In case of myoglobin, it is the histidine residue. but here it is the cysteine residue. Then we have discussed its mechanism, different kind of reactions that has been shown by cytochrome P450, and its mechanism of action, how it acts on the substrates. So this is all about the cytochrome P450.